a 2-week hiatus, and would like to kick off everything today with a bang with Professor Michael Georgieff from Minnesota, where it's still cold, snowing, and miserable. So thank you, Michael, for joining. I met Michael first time in India, where he blew me away with this talk about iron. It's something we don't think about, right? We're all talking about oxygen and this and that, but we forget about diet and how important it is to the neurodevelopment of children. So as usual, a few housekeeping rules. Please keep yourself muted and type questions for Michael to answer at the end of his talk. And if really desperate, put up your hand with the hand raise sign and he's sure to pause for you to ask, but hopefully we can make it to the end. This session is being recorded and we'll check it for any dodgy bits before we post it. And also remember that each one is accredited for 1 hour of CME points with the Physicians College. And if you're with another college, ask them what it's whether you can use it. All right. Okay. I am busy admitting everyone, Michael. So I think the floor is yours. Thank you very much.
Thank you, Julie. Yes. Hi, everybody. I'm going to share my screen and we can get started here. There we go. So hopefully you see a title slide that says iron and brain development. And
yep.
Yes, I'm coming to you from the university. Well, I'm coming actually from my house because it is after 9:00 p.m. here. And I'm not on call, which is great. So I am actually at home. And but I work at the University of Minnesota and I co-direct something called the Masonic Institute for the Developing Brain. I'm a neonatologist by training, and I work in what we would call nutritional neuroscience. And because we now know, and as we will be talking about, now we know that fetal nutrition is very important in terms of setting up postnatal nutrition. I'm also appointed in the Department of Obstetrics and Gynecology and cross-appointed into the Institute of Child Development, which is the child psychology department. So way too many hats. I didn't ask for most of those, but that's, I think, what comes with the territory. Our objectives here are to talk about iron, right? But we want to think about iron actually as a paradigm for other nutrients that affect brain development. Remember, nutrition is one of the things that we can actually modulate in the NICU that has an effect on developmental outcome. We're not very good at modulating whether a kid has a hemorrhage or not, or whether they have hypoxia-ischemia or not, but we do have the ability to modulate things like your level of anemia in the unit or your nutritional status. And we'll talk about why that's important. So iron will also then therefore stand as a paradigm for other interactions of other nutrients with the brain, or for that matter, other environmental factors, for example, stress in the brain or something like that. We're going to talk about, we'll hope that you can define the maternal risks to fetal iron accretion, because then this is kind of a spoiler alert. You don't just start with a blank slate when you're born. You have a fetal history and a fetal nutritional history that comes with you, both in terms of the nutrient itself, but also how it's influenced brain development up until the time of birth. Then we'll talk about what happens to the iron status of preterm babies in the hospital and the iron requirements of those babies. And then the story is not over when we discharge the babies. I've run a NICU follow-up clinic for the last 35 years, and I can tell you that the nutritional requirements continue. And if you need to be convinced of that, just ask yourself, when you send home a baby from the NICU, do they look like a term baby or do they not look like a term baby? And of course they don't. And that reflects some of the nutritional issues that they're having. The reason this is important, and the reason I got into this a very long time ago when I was a neonatal fellow, is that we began to realize in the 1980s and 1990s that babies were not born with a blank slate. In fact, they came with, like I said, a history. And about that time, the Barker hypothesis, or what became known as the developmental origins of health and disease hypothesis, was becoming popularized. This is a cover from Newsweek in 1999. And I think there are two things that are quite interesting here. The one we're going to talk about, which is where health begins and how your risk of obesity and cancer and heart attacks are they say set in the womb. That sounds very deterministic. It's not, but your odds are changed based on what your intrauterine environment was like. In 2023, and certainly we know for the last probably 15 years, mental health should have been on here as well. That is, the programming of brain development and therefore brain function across the lifespan does take place during fetal and early neonatal life. So we are in the midst of that when we are taking care of babies who are spending their third trimester out in the NICU, or if you're an obstetrician, taking care of these moms as they progress even toward a normal term delivery. Of course, the other interesting one here, and I'm sure is near and dear to your hearts in Australia, is get ready for more storms like Floyd. So Floyd was a huge hurricane in the U.S. in the 1990s, and this was indicative that something was happening with our climate change and so on. Of course, both of these issues, so true back in 1999, are still true in 2023. And the reason that's important is because these early years, basically what people would call 0 to 3, or probably more accurately, the first 1,000 days, which is really from conception to about 2 years of age, is a time when some primary systems are developing in the brain that are absolutely essential to kids being able to learn and develop and thrive in school. So for example, the learning and memory system, based primarily in the hippocampus and striatum, is rapidly developing, ramping up around 28 weeks gestation, and then going on postnatally until about 18 months to 2 years. Your hippocampus actually is constantly renovating itself, but the rapid development time, the time where it would be most at risk for nutrient deficiencies, starts at about 28 weeks gestation. Similarly, myelination rapidly ramps at about 32 weeks gestation. So again, it's the time that we have the kids in the unit, and proceeds at a rapid or brisk pace for a couple of years. Again, you're constantly turning over your myelin, but again, the critical period would be early in life. And then something that I don't think is well known, and that is that your reward systems, so for example, the neurotransmitters that are involved, like dopamine and serotonin and norepinephrine, start being shaped at about mid-gestation. So these are primary systems in the brain that are sensitive to extrinsic influences like nutrition, stress, and so on, that are being shaped by those and patterned, and as I will show you, for the lifespan. But not only are they important in developing then, but later developing systems, ones that are not developing at the time that we have them, for example, in the NICU, or maternal fetal people might have them in utero. Later order systems like the prefrontal cortex are dependent on the fidelity of those early developing systems. So think of the brain as being very regional and being scaffolded in terms of its development. So these primary systems will eventually connect to the prefrontal cortex. And one of the indications that that happens is that many of the preemies that I see in NICU follow-up clinic have attentional problems, inhibition problems, working memory problems. Those areas are not particularly developing at the time we have them in the unit, but they're dependent on these primary areas that are. That kind of leads to the next point, which is, you know, if we have nutritional disorders like a baby's not growing well, or has stunted linear growth, or is iron deficient, and you give them the nutrient back and they show you catch-up growth and catch-up in their nutrient status, and everything returns to normal, then we don't have a huge problem. We can always rely on catch-up. So an organ like the brain can be affected only during the nutritional alteration. We call that acute dysfunction. So it shouldn't surprise you, for example, that a iron deficient brain doesn't work very well. What we get concerned about is the effects that last beyond the time of the alteration. In other words, you make the diagnosis, you correct the problem, and yet there is still an increased risk of adult dysfunction and risk of mental health problems. And we're going to talk about what the mechanisms are, because they are known, what the mechanisms are behind these long-term effects. That's your cost to society. I have just a quote that I like from the Lancet back in 2008. If we eradicated the three most common micronutrient deficiencies, so iron is the most common, zinc deficiency or iodine deficiency, we'd increase the world's IQ by 10 points. And so it shows you that there are these long-term effects. 10 points in IQ makes a big difference in terms of what you do for a job, what your educational potential is, and so on. So you can kind of see how this becomes a transgenerational thing. If you don't fulfill your potential in one generation, it's going to pass on then to the next one. Now, why is that the case? Well, that's because there's an enormous amount of brain development that takes place in that last trimester. So this is what the baby's brain schematically looks like when we take care of these kids. We resuscitate babies, at least at my unit, down to about 22 weeks gestation. And at that point, the brain looks like basically a coffee bean, right? Bilobed, smooth, no sulci, no gyri. And by 9 months, not 9 months postnatal age, by 9 months post-conceptional age, so the term brain down here in the lower right looks much more like an adult brain. And that's because of all the gyrification and sulcation, which is, of course, Mother Nature's way of packing a lot of volume into, or a lot of surface area into a small volume. You can think of this also by looking kind of internally, what's going on inside. So I think the first thing to think about is that the brain is not a single organ. We refer to it in terms of brain development, or this has an effect on the brain. But the brain, talking about the brain like that is like talking about the thorax. There's lots of things inside the thorax. Yes, they intercommunicate, but they are all distinct organs. Same thing with the brain. You have different regions. I talked to you about hippocampus. You have different processes like myelination and neurotransmitter synthesis, and they're all on different developmental trajectories. So what I've done here is I've taken a slide that my good friend Chuck Nelson published back in 2001 showing human brain development across the lifespan. And the first thing you'll notice about this is the tremendous stretching of the x-axis until the age of 3. That literally takes up half of this graph. And that is to illustrate all the waves of activity, and this is designed to show you when the peak development is of these various areas, many of which are taking place before the age of 3, many of which are actually starting well before the time of birth. So I've shown with red arrows here those processes that start before term birth and therefore would be at risk either for maternal fetal complications or NICU complications. And then you notice that the rest of life is just kind of compressed into this right-hand side over here, where the decades just kind of fly by without much brain development. And unfortunately, you do have neurogenesis in the hippocampus across the lifespan, and you do make synapses, so you are able to learn. But for the most part, brain development to a great extent is quite well done by the age of 3. Of course, the frontal lobe continues to develop across the teenage years and probably into the early 20s. So again, think of these as waves of development and that they are dependent on the integrity of the previous stage. So why is that early brain so at risk for nutritional perturbations? Well, as you and all are sitting and listening, and I have an excuse because it's 9 o'clock here at night, but assuming that everybody there is awake, your brain is using about 20% of your total body oxygen consumption. And your total body oxygen consumption, or your total body caloric needs, let's call it that, is about 40 to 45 calories per kilogram of body weight per day. Now, you all, I'm sure, know that a baby has a much higher oxygen consumption rate, and its total caloric needs are around 100, if you're a preemie, maybe 120 calories per kilo per day. So you've already got a metabolic machine in that baby that is revving at about 2 to 2.5 times what it would as an adult. And as the slide says, 60% of the baby's energy goes to the brain. 60%. That is the cost of growth. And what growth is that? Well, that's dendritic differentiation, synaptogenesis, the brain connecting itself, becoming more complex, like I showed in that slide a couple of slides ago. So then you can ask the question, all right, well, what nutrients, let's call them metabolic substrates, support that type of big-time oxidative metabolism, that big-time generation of ATP? And the answer is, of course, oxygen and glucose and amino acids, the ones that we're always checking on rounds. But they also include the micronutrients like iron, because iron is in cytochromes, and cytochromes are absolutely crucial for electron transport and ATP generation. Similarly, copper is there basically to get iron into the brain. And then iodine, which is only in the brain or only in the body to make thyroid hormone, which sets the metabolic rate and the metabolic rate of your brain. So these are the substrates that we worry about when they are short-changed, that will reduce the rate of brain development. So I highlight iron, of course, because that's what we're going to end up talking about here. Iron is a good paradigm for how a nutrient interacts with the brain, because its biology is extremely well known. It's one of the most studied nutrients out there, and we know what it does. It's found in these clusters, either as hydroxylases or hemoproteins like hemoglobin, myoglobin, neuroglobin, and so on. So they're important for tissue oxygen delivery. I think we all know that. But they're also involved in very specific aspects of brain development and function. So, for example, myelination, which I told you starts ramping up at about 32 weeks gestation. Multiple desaturases that are there to generate the fatty acids in the coat that are part of the myelin, of course, that coat the nerves, are iron dependent. The structural development is highly dependent on energy production by the mitochondria through the electron transport chain. And again, cytochromes are critical to that. Iron is critical for that. So think in terms of dendrite arborization and synapses. The wealth of dendritic arborization, or how well the arborets look, relates directly to the capacity. So the more simple dendritic structure is going to be a less capable system in any of the regions of the brain. We talked about neurotransmitters. Well, the monoamines are directly synthesized by enzymes containing iron. So both tyrosine hydroxylase and tryptophan hydroxylase contain iron in their enzymatic pockets. If you are iron deficient, you alter your dopamine metabolism, your serotonin metabolism. And then more recently, mostly our group, but others too, have found that iron regulates long-term gene expression through epigenetic modification, particularly to synaptic plasticity genes like brain-derived neurotrophic factor. That's a growth factor for the brain. And they do it again through iron-containing enzymes, either in demethylases when we're talking about histone methylation and demethylation, and also in DNA CpG hydroxymethylation. Those are two iron-dependent processes. And so we know that early iron deficiency, and I'll show you the data shortly, affects how the chromatin in the brain is epigenetically modified, and it's modified across the lifespan, at least in our mouse and rat models. So again, this whole idea that better to do it right in the first place, i.e. stay nutrient sufficient, iron sufficient, as opposed to trying to rely on catching up where you may not have the opportunity anymore. Right. So now we go back to our human brain development slide, and I've just circled some of the processes that are starting prior to term birth and that are iron dependent. Remember, we talked about synaptogenesis, myelination, and rapid hippocampal development. That's structural development. Now, before I get too much further, everybody I know knows that iron is also a potential risk. It is a potent oxidant stressor. I remember I was giving a talk once and somebody said, oh, this was at PAS, Julia, and they said, here's Mike Georgieff. He's going to talk about iron, the bad boy of nutrition. And indeed, it is kind of the bad boy of nutrition. So it is very tightly regulated for a good reason, because both deficiency and overload are both quite toxic to the brain. Iron overload has been associated with neurodegenerative disorders, including Alzheimer's and Parkinson's, and also after reperfusion injuries, after acute stroke or hypoxia ischemia. This is in adults. Actually, you can see that in neonatal models as well. Now, I want to emphasize that's probably all not nutritional iron. That's probably leakage, and probably has little to do with what your dietary iron is about. But for our fetuses and our premature infants, they are theoretically at high risk because they have underdeveloped antioxidant systems. They also have low protein levels, right? And so iron is only dangerous if it is non-protein bound. It's not supposed to be non-protein bound. It is usually bound to transferrin. That's what makes up most of the total iron binding capacity. And babies, and particularly preemies, have low levels of transferrin, low levels of total iron binding capacity. So there's potentially an opportunity to overwhelm that binding capacity and create potential for oxidative diseases like retinopathy or BPD or necrotizing enterocolitis. So there is concern on the overload side as well. You know that nutrients basically follow a traditional U-shaped risk curve where your lowest risk is in the adequacy range. And both overload and deficiency can cause issues. And what I've drawn here in purple is to show you that iron, the stakes are just a bit higher, that you get into trouble a lot quicker. So why worry about iron deficiency? And the answer is because 2 billion people of the 7.5 billion of us that there are on this planet are deficient, and in particular, pregnant women and their children. Since every cell needs some source of energy and is generating ATP, basically every cell and every organ system needs iron for proper development and function. Now, most people, most probably hematologists, if you ask them why you have symptoms from iron deficiency, they will say, well, it's because you're anemic and you have reduced oxygen delivery, and that's why you feel tired. But in fact, the symptoms occur prior to anemia. And the reason you feel poorly with iron deficiency or have poor brain function is due to tissue level iron deficiency. That is compromise of, in the peripheral tissues, for example, muscle, myoglobin and cytochromes, and in the brain, compromise of the cytochromes, the myelin, and the neurotransmitters, and so on. And you can show that easily in an animal model by making them iron deficient anemic and then transfusing them, getting their hemoglobin back up, and they still will have all of the symptomatology of iron deficiency. The main reason we worry about iron is the developing brain, because the effects appear to be, some of the effects appear to be permanent, meaning, yes, your brain does not function very well when you're iron deficient, but, there are both cognitive and motor effects of that have been described. But many of these problems last long after iron repletion. Some of the studies now are out 30 years showing that the people who were iron deficient as young children continue to have issues with anxiety, depression, poor mentation, poor working memory. So we need to worry about this. All right, I think the big paradigm shift that has taken place in the last 10 years or so has been that iron deficiency that we are all taught about in children, so the toddler that is iron deficient, used to be assumed that was simply due to not eating enough iron and not having enough iron in your diet, combined often with intestinal blood loss, say from parasites and so on. What we've learned in the last 10 years, actually from a study that was done in China, by Betsy Lozoff and her Chinese colleagues, is that how soon the baby has, or the child is at risk for postnatal iron deficiency has a lot to do with how well they were loaded as a fetus. And so just like we found out with brain development, there's a continuum from prenatal to postnatal life. So I think it's important to know, what are the risks for iron deficiency at birth. Now, mind you, very few of those kids at birth are anemic, but there are gestational conditions that result in the tissues being iron deficient, even though the kids are not anemic. And that illustrates a basic principle that I want you to know, which is that iron is prioritized to red blood cells. All right. So when you are dosing kids with iron, it is going to build, first thing it's going to do is go to the red cells to build back the hemoglobin mass. And, of course, that's important for oxygen delivery, but it does so at the expense of the brain, at the expense of the heart, at the expense of skeletal muscle, and certainly at the expense of the storage pool, which is found in the liver. Pregnancy has been described as an impending iron deficiency state. So, if you were going to ask the question, when would you intervene? Ideally, we would like to have women entering pregnancy to already be iron sufficient, because here's what happens when you become pregnant is the mother expands her blood volume. So that's going to take more iron, right? You need 3.46 milligrams of iron for every gram of hemoglobin synthesized. And then there's the placenta, which is a very active metabolic organ and has high iron demand for itself. And then you've got that fetus at the far end, who also has a high iron demand for blood, organ growth, and for storage. So iron deficiency rates in some countries can approach 85% if you use sensitive enough tests, for example, percent transference saturation or ferritin or something like that. In a study that we're currently doing in India, 67% of pregnant women in certain parts of India are iron deficient. And India actually has a program of what they call anemia mukht bharat, which is to wipe out iron deficiency in pregnancy through iron and folic acid supplementation, deworming, point of care testing of anemia, delayed cord clamping for the baby, and ruling out other anemia causes. So very high rate, exacerbated then by a woman becoming pregnant. The fetus, in the meantime, sits there and is accreting iron. And another basic principle is that the fetus is actually quite good at taking iron from the mother, right? So the transport mechanism is actually the fetus calls for the iron through its own transferrin saturation, signaling to the placenta, hey, I need more iron over here. That's great up to a point, but once the maternal fetal dyad, once the mother reaches a certain degree of iron deficiency, then the entire maternal fetal dyad becomes deficient. So basically, fetuses during the third trimester have about 75 milligrams of elemental iron per kilo of body weight. And what that means is they go, as a 24-weeker, they might have about 37, 38 milligrams of iron. By the time they are term, they will have over 200 milligrams of iron. So a lot of the iron is accreted in the last trimester. And I want you to think about that, because when we have babies who have missed their last trimester and in our NICUs, not only are they not getting the iron they would have gotten in utero, but we are taking it away from them through phlebotomy. And we're going to go through some of those stressors in a second here. Most iron, just like in adults, is found in red cells. But one thing that is very different about fetuses and newborns is that they have pretty large iron stores. Those iron stores are actually to be utilized in the immediate first 4 months postnatally, because breast milk actually has very little iron in it. So in order to support the expansion of the blood volume postnatally as the baby grows, to support brain development processes and so on, it actually, the newborn actually utilizes those large iron stores. And it's when those stores are compromised by certain gestational conditions, or by us when we phlebotomize the babies and they use that iron, whatever iron they do have in their livers for erythropoiesis, that the non-storage tissues, including the brain, which is the smallest compartment, gets compromised. So again, take-home message here is that fetal and neonatal iron is prioritized to red cells over the brain. So non-anemic infants can be brain iron deficient, and we've done autopsy studies to show this. And they are detected by having low iron stores, and that is a ferritin less than 76 micrograms per liter. I mentioned earlier that the fetus is real good at taking the iron from the mom, but there are gestational conditions that are associated with iron deficiency and that also cause iron deficiency. So First of all, of course, maternal iron deficiency. There is a break point or an inflection point, and I'll show you this graph in a second, of a maternal ferritin level less than 13.6, at which point she is depleted enough that it starts to affect the fetal iron stores, or a hemoglobin less than 100 or less than 10.0. Iron deficiency is also induced by intrauterine growth restriction. When you look at a growth restricted baby, you're looking at protein energy malnutrition. That's what you see with your mind's eye. But over 50% of intrauterine growth restricted babies have compromised or low iron stores. Premature delivery increases the risk of iron deficiency in young children. Why? Because they didn't get to accrete that iron in the last trimester. I think it's important to recognize that both IUGR and premature delivery are increased in incidence in moms who had iron deficiency anemia in first and second trimesters. So it's kind of a compounding effect there. And I'm not going to talk about this particularly, but it turns out that infants of diabetic mothers also have very low iron, tissue iron status. And that is mostly due to the fact that they've got a huge red cell mass, right? They're often very polycythemic. So all that iron is stuck in the red cells. Every one of these conditions is associated with poor neurodevelopmental outcome. So that tells you about the bright iron deficiency. When we look at the relationships between maternal iron status and fetal iron status, look up here in the upper right-hand corner, and you see, first of all, that there is a lot of scatter in cord blood serum ferritin levels, but that maternal iron deficiency, once she has a serum ferritin less than, as I said, 13.6, then you see compromise of the stores in the baby. Betsy Lozoff, who I mentioned before, looked at iron status of infants at 9 months. Again, this study was also done in China. Infants, what their 9-month iron status was most dependent on, and yes, things like what you feed them and postnatal iron supplementation makes a big difference. But everything you see in yellow on here is a prenatal factor that is influencing the iron status of the children at 9 months of age. So again, it emphasizes the importance of adequate fetal loading. We have some estimates of how iron deficient these kids' brains are. Now, your typical toddler with iron deficiency, obviously we don't know how deficient they are. But babies occasionally with intrauterine growth restriction don't survive. Occasionally infants of diabetic mothers don't survive. And we've used autopsy approaches to look at the degree of iron deficiency that those conditions induce in the brain. And you see that it drops the iron by 32 to 40%. That's important because that's actually what our models, like the one I'm showing you down here in the green, we can model that degree of iron reduction in the brain. And what you're looking at here in the green slides is an iron sufficient brain kind of in the middle, and an iron deficient brain, or a formerly iron deficient brain, out here on the right. These are adult brains in mice that were iron deficient only in the neonatal period. And you can see in the iron sufficient animal, this is the hippocampus. The cells, the neuronal bodies look really pretty. You don't have to be a neurobiologist to know that this is a very organized looking brain with very nice looking dendritic tree. And the iron deficient brain, in contrast, is very disorganized with early breakpoints and very abnormal branching. And not surprisingly, these animals perform rather poorly on cognitive and memory tasks. So it kind of puts a picture on what a similar degree of iron deficiency might be doing in a human brain. Is there evidence that there are long-term and short and long-term neurobehavioral effects? Yes, there are of neonatal iron deficiency, yes. So acutely, these babies have poor recognition memory. We are actually able to test recognition memory in absolute newborn babies with and without iron deficiency. And those with iron deficiency have poor discrimination of stimuli. They have slower speed of processing. So you can do ABRs on these kids, and you will see that their ABRs are slower than kids with normal iron status. And probably the monoamine effect is seen in this paper by Ted Wax, where he showed poor maternal-child interaction and poor bonding. From a fetal standpoint, first trimester iron deficiency increases the risk of autism in the offspring. Second trimester increases the risk of schizophrenia. And third trimester iron deficiency increases the risk of neurocognitive problems. So you can kind of see how timing is very important, and it probably has to do with what iron-dependent circuits, neuronal circuits, are developing when the iron deficiency happened. And finally, if you are born with low stores, you're going to, as I said, have an earlier onset of postnatal iron deficiency. Those low stores are depleted earlier, and postnatal iron deficiency has been associated with motor dysfunction, socioaffective disorders, risk of depression and anxiety, all the way into adulthood. So there's plenty of evidence, and there's probably plenty of good biology for us to understand why iron is necessary for that developing brain, either as a fetus or a neonate. Does supplementation work? Should we be supplementing people? Well, you should be supplementing anybody who's iron deficient. Prenatal iron supplementation in populations that are at high risk for iron deficiency improves the pregnancy outcome, meaning there's less prematurity and less IUGR if you supplement them in the first and second trimester. And probably the most compelling study was done by Perul Christian, where she randomized moms to either get iron supplementation or not, and then looked at the outcomes of the kids. And at school age, the children that were born to moms who were supplemented during pregnancy had better neurodevelopment, better school age performance. What was compelling about this was that she took the group that was randomized to placebo during the prenatal period and re-randomized those kids once they were born to iron supplementation or not, and it didn't make any difference. So it tells you that the fetal or the post-conceptional time period up to term is whether they're with us or they're in utero, seems to be a really important time in terms of outcome. Postnatal iron supplementation certainly improves hematology. There was a New England Journal article about this recently, but it has variable effects on neurodevelopment. If you are iron deficient, or if you're in an iron deficient population, Betsy Lozoff's work, Fenji Geng, who was her collaborator in China, all showed a positive effect of postnatal iron supplementation. On the other hand, supplementing people who are already sufficient, there's not a lot of evidence that that improves things long term, and may be in fact detrimental. All right, returning to our babies. So their iron, let me just see what's in the chat here real quickly. It's a CME thing. All right. For our babies in the unit, there are a lot of stressors, a lot of push and pull on the iron status. So let's first set the stage here and say that, first of all, preterm infants, and that's what I'm showing you up here in the upper left, preterm infants are generally born with lower ferritins, therefore lower iron stores than babies born at term. So just for measurement, if you're measuring serum ferritin in the unit on your kids, the average ferritin for a term baby is about 134, and for a preterm baby is a little bit less. As I said, we see neurologic problems somewhere around the 20th to 25th percentile mark, meaning with ferritins less than 76. What are the factors that determine iron balance in the unit? Well, I've tried to juxtapose them here, right? One thing that would send you into negative versus the thing that would send you into positive. So low endowment or earlier gestation tends to put you in negative iron balance, whereas being of older gestation and being appropriate for gestational age tends to keep you in positive. Phlebotomy. So my good friend and collaborator, Dr. Jack Widnes, said we should stop calling it anemia of prematurity and just call it anemia of phlebotomy, or phlebotomy-induced anemia, because that indeed is the biggest iron loss that our babies undergo. There are estimates that babies may have their entire blood volume removed, certainly during the hospitalization in the sicker kids, as often as every 2 weeks. Now, we do give them red cell transfusions, and that's a lot of iron. But remember that we're always phlebotomizing first and transfusing back. Rarely do we have babies whose hemoglobins are what they would have been in utero, or even what a term baby has, say 14 or 15. We're running kids with hemoglobins of 8, 9, 10, 11. So they are always behind the 8 ball as far as that goes. In the US, the American Academy of Pediatrics recommends starting iron therapy anywhere between 2 weeks and 2 months in preemies. If you start it as late as 2 months, you will have about a rate of 69% iron deficiency in the follow-up period. The AAP also recommends iron at a dose of 2 to 4 milligrams per kilo per day. And if you have kids that you're treating with recombinant erythropoietin, that number will go up to 6 milligrams per kilo per day, and potentially even higher. Then that's because you need to fill all of the buckets, and you're pushing the iron into the red blood cells with EPO. In order to protect the brain and protect the liver stores, you're going to need to give them at least 6 per kilo per day. And then something that we consider very good in the unit, which is rapid growth, actually is a real stressor on the iron system, because with rapid growth comes expansion of the blood volume, and obviously you need iron to do that as well. Coming back now to one of our main causes for iron deficiency in the unit, especially brain iron deficiency, we've been able to model phlebotomy-induced anemia in mice by dropping them to hematocrits that we see in the NICU all the time, 25%, that's not an unusual hematocrit. And when you look at the brain iron of those animals, they have a reduction in brain iron of about 40%. And I've already shown you what that does to the structure of the brain. And again, that's because any dietary iron, in this case, that these animals are getting, is being put into the red cells to try and get that hemoglobin back up, or that hematocrit back up to where it should be. And what gets sacrificed then is the brain. Does that make a difference? Well, yes. Here's a graph that shows you relative to control non-bled mice. You drop the brain iron by 40%, and you literally double the lactate in the brain. So there's your evidence that likely effects on cytochromes and degeneration in oxidative metabolism. Does that make a difference in terms of expression of synaptic plasticity genes? Yes, BDNF, brain-derived neurotrophic factor 3, is reduced substantially, as is something called postsynaptic density protein 95. So these are important genes in terms of making the brain work well. We've, in neuronal culture, and I'm just going to briefly talk about this, we have looked at the effect of iron deficiency in neurons on oxygen consumption rate. So up here in the upper left-hand corner, in black, you see the normal oxygen consumption rate of these cells, and you can see it's literally halved in the red lines, literally halved by a 40% reduction in brain iron, or in neuronal iron. Does that make a difference in terms of structure? Yes, if you look at the panel here with kind of the robust picture of lots of dendritic arborization, that's the iron sufficient. In contrast, the one that's marked C is your iron deficient animal. So it makes a difference in terms of structure and function. So what about those long-term effects? Well, there's 2 theories behind that. The first is what's called the critical period hypothesis. So I showed you how there are these waves of development in the brain, and they require that those time periods of rapid development, critical periods, require substrates. Iron is one of them. If you have insufficient substrate during that critical period, you will affect what the brain looks like, how it builds itself. And if the critical period closes, which it does, any structural disorders will remain into adulthood. And the neurobehavioral deficits that you see, for example, poor learning and memory, is directly related to that change in structure. The second, kind of more interesting, to me at least, effect is what I mentioned at the very beginning of the talk, which is that iron is known to change the chromatin structure, so epigenetic modifications. And by the way, This is not unique to iron. There are a bunch of nutrients that alter brain epigenetics, and particularly of synaptic plasticity type genes like brain-derived neurotrophic factor. So for example, the fish oils, which everybody thinks are important for brain development, well, indeed, they do change the DNA methylation of BDNF. Intruding growth restriction changes hippocampal methylation, histone methylation. Choline appears to positively affect the brain in terms of chromatin and in terms of improving neurodevelopmental outcome, and is being used in conditions like fetal alcohol syndrome and Down syndrome to try and improve developmental outcomes. It is a methyl donor and is thought to be involved both in CPG and histone demethylation. And then iron, which I'm about to show you here. So remember I told you that brain-derived neurotrophic factor, important synaptic plasticity gene, and the reason for that is here's what's downstream of BDNF in various brain regions, and those proteins that you see down here are involved in kind of important things like cholesterol synthesis, which makes for synaptic vesicles, neuronal activity, long-term potentiation, how much electricity your brain can make, glia and oligodendrocyte development, all of which kind of play together as neuroplasticity. What I'm showing you bar graph wise down here are animals that are either have been always iron sufficient, they are marked IS and are in the dark bars, or formerly iron deficient. So these are adult animals that were iron deficient only as fetuses and newborns and then repleted. And you can see that their brain-derived neurotrophic factors remain suppressed even though they've been iron sufficient for months. These various other genes and proteins also permanently suppressed. So you can imagine that that does not lend itself well to behavioral activity. One of the areas that Phu Tran, who is one of my colleagues at the University of Minnesota, has explored is this business of iron-dependent demethylase, histone demethylase. It's called GERDs. And here he's looking in the adult. So again, these are formerly iron-deficient animals, now in the dark bars, and looking at marks of methylation of histones. Now, you need to know something about these. These are histones that are regulating the BDNF, helping regulate the BDNF gene. K27 is a suppressive mark. And so you see permanent increase in the amount of methylation of this suppressive mark on BDNF. K4 is an activating mark, and you see suppression of that. So don't get confused by the double negatives, but this is a way of showing that a pattern that occurred in early life has now been propagated all the way out into adulthood and is changing how this gene is being expressed. You can also look, and I think this work is interesting because it ties it back to the human. You can also look at gene networks. So you can use broad approaches like RNA-seq to look at differences in gene expression in adulthood between iron sufficient, always iron sufficient animals and formerly iron deficient animals. So again, animals only deficient in the newborn period, now sufficient as adults, and kind of divide these up, if you will, into 6 lanes apiece. And you can see that genes are really very differentially expressed in the formerly ID compared to the iron sufficient. You can take that information and crunch it and say, okay, well, what are these genes? What do they code for? And here's what they code for. They code for autism, they code for schizophrenia, and they code for Alzheimer's. And I find particularly interesting the autism and schizophrenia because that's exactly what the human epidemiology studies showed too. That remember, first trimester iron deficiency, greater risk of autism in the offspring, second trimester iron deficiency, greater risk of schizophrenia. So it's kind of a neat way of showing that the epidemiology was right. So all of us sit here and go, all right, the conundrum is what's an acceptable hemoglobin for a baby in the hospital? We talked about how we pretty much remove the entire blood volume every 2 to 8 weeks. And so where do you keep your hemoglobins and what does the literature tell us? There are really 3 randomized trials that have looked at what hemoglobin should you be defending? When should you be transfusing, a high threshold or a low threshold? The first one is what's called the Iowa trial, pretty small, single center trial. Really didn't show much of anything other than a little bit more apnea and IVH in the low threshold baby. So that would have suggested that anemia is not good for the brain, and that shouldn't surprise anybody. But the Bailey scores were really no different between the low threshold and the high threshold babies. But they brought some of these kids back later, and here they found, particularly in girls, that some of the less anemic kids, in other words, ones that got more transfusions, and particularly females, had greater problems. And these were some of the areas that they had problems in: verbal fluency and visual memory. Now, they didn't use EPO, so I think the question was it the transfusions? There was a multi-center Canadian trial that randomized 451 kids, so a little bit bigger, and they really saw no differences in long-term follow-up neurodevelopment between a low threshold or a high threshold for the transfusion. And you probably all know about the TOP trial, which was published a couple of years ago in the New England Journal, that showed now an even bigger group, 1,692 kids. And again, no difference in outcomes, either IVH or neck or retinopathy or long-term or neurodevelopment beyond 2 years of age. And I think the important thing here is that every baby got transfused in the study. So if it only takes one transfusion to torch the brain, then I'm not sure that there really is a difference between 6.2 and 4.4. This just shows you that the difference in hemoglobin actually wasn't all that large between the groups. And why am I telling you that? Because I don't think you can tell from that whether phlebotomy-induced anemia without therapy alone, just where you keep them anemia-wise, which all the models would suggest would be bad for the brain, you cannot tell from this trial because most of these kids were transfused, or almost all the kids were transfused. It's hard to tell what the effect of erythropoietic agents are, like darbepoetin or EPO, because those kids are transfused as well. And so it's hard to reach really any conclusion on the effects of either treatment or the degree of anemia. Neurodevelopmental studies have been done just as a function of iron deficiency in preterms, and that literature gets bigger and bigger. A couple papers published in the last couple of years show improved Bailey scores related to cumulative iron dose that they got in the NICU, and improved Griffith scores if kids were supplemented with late preterms were supplemented with iron or not. This is data from the PEANUT trial that shows that cumulative iron dose, either the top three are at day 60, the bottom three are at day 90. We're looking at Bailey scores, either cognitive, motor, or language. And in all cases, you see cumulative iron dosing improving outcomes at 2 years of age. And if you're wondering the difference between the black line and the dotted line, the black line are the placebo kids, the kids that did not get EPO. So there was no pressure to put it into the red, as much pressure to put it into the red cells. The EPO kids had a slightly lower slope. And in Sweden, Stefan Berglund and Magnus Damelhoff asked the question, what about late preterms, kids born between 2,000 and 2,500 grams, either because they're premature or they're late preterms? They were randomized to either get no iron at all, just breastfed, no iron, or breastfed with 1 or 2 milligrams per kilo, and they were treated for 6 months. Much higher incidence of iron deficiency anemia and iron deficiency without anemia in the kids that were not given iron. So that tells you that late preterms, even though they look like they are term, they have not accreted all of their iron, and that they deserve treatment with iron. Does it make a difference? Yes. When they brought those kids back at 3 years and again at 7, there were a lot more problems with emotional reaction and attention problems in the kids who got placebo, 13% right there, compared to those who actually got treatment with iron. After discharge, it doesn't end. We typically discharge kids at, say, 2 to 2.5 kilos. They are close to term. A normal term baby is about 3.5 kilos. We send them home with hemoglobins, at least from our unit, somewhere in the range of 7 to 11, whereas a term baby would be going home with a hemoglobin of 13, and their ferritins are really all over the place. what does that mean? That means that you want to be dosing these kids with about 2 per kilo of iron after discharge if they are leaving with a normal hemoglobin and a normal ferritin. But if they are leaving with a low hemoglobin, let's say hemoglobin of 8.5, and a ferritin that is low, say 40 at the 5th percentile, that number rises to 4 milligrams per kilo per day. And we generally advise that that be done for at least 6 months of age post-discharge. I'm going to spare you this. I mentioned this before, that if you start iron late, you're going to have close to a 70% rate of iron deficiency in the follow-up period. A Cochrane review of enteral iron strategy prior to discharge in the NICU, does it affect post-discharge iron status at 1 year? There are 26 studies that have looked at that. And once again, the conclusion was 2 to 3 milligrams per kilo per day in the discharge period is adequate. So where does that leave us? That leaves us with this kind of dosing scheme. Term babies who are on formula are going to get about 1 milligram per kilo per day. In general, breastfed babies may well not need much iron, at least not in the first couple of months. And why give them something that they don't need? They have adequate iron stores if they are born appropriate for gestational age and had delayed cord clamping. Babies who are in the 2,000 to 2,500 gram range, either because of being IUGR or premature, need 2 per kilo per day. If you're less than that, as I just showed you, probably 2 to 3 milligrams per kilo per day. When we check ferritin levels on our babies in the unit every 2 weeks, and when they have a low serum ferritin down in that danger range below where we think neurologic effects happen, we increase the dose by 1 to 2 per kilo per day. If kids are on EPO, they'll typically get 6 per kilo per day. And of course, there are those kids who get multiple transfusions for blood pressure support, and they have high ferritins, and we wait until the ferritin comes down to restart their iron. So we stop their iron. So let me summarize, and then maybe we can have some questions. I hope I've shown you that iron has an important role in brain development. We talked about myelin, structural development, monoamines, genes. It's regulated in a very narrow range. We don't know how effective preemies are in that regulation, and that's an area of study. Iron deficiency certainly presents a risk to the developing premature brain. We have plenty of studies on both the effect of iron deficiency and the positive effect of supplementation on their developmental outcome. But iron overload is a very underexplored area. A lot of hand waving about it, a lot of concern about it, not many studies done about it. You need to remember that the post-discharge preemie is at risk for iron deficiency, especially if they're growing rapidly, which they are. And it's something I didn't talk about, but one of the reasons we are hesitant about iron therapy in term breastfed babies is that unabsorbed iron can shift the gut microbiome toward more pathogenic strains. But that's not been studied in preterms, and it's an area that I think will be very interesting. So I will stop. I always have to say that I go into my laboratory and they say these days, you're not going to touch anything, I hope. And I don't. These are all the people on the left here who really do all the great work, and my funding sources and collaborations really around the world that have made this a fun area to be in. So I'll stop sharing here, and let's see if we have some questions.
Thank you so much, Michael. Amazing talk. Should we give women who are at high risk of anemia during pregnancy, like the diabetics, more iron? Is that normally done in your practice?
It is not normally done in our practice. My daughter was recently pregnant and was going to the clinic that I work at, or the hospital that I work at. So she asked me if I would come to the clinic visit with her. And the the maternal fetal medicine fellow came in, and I asked the fellow, so are you going to check her iron status? And I was told, oh, no, sir, we don't do that routinely here. It's like wrong answer. It is not. It is not commonly done. And there have been articles written, and I've written one of them, but there are more and more articles showing up that are saying, one, we're missing an opportunity both for the moms in terms of outcomes, less IUGR, less prematurity, and also for the brain development of those kids, if we're not screening moms at the beginning of pregnancy and doing something about it. Oral iron therapy, as everybody knows, is a bear. I mean, people don't like to take it. The compliance is very low. So there are people who are studying whether IV iron might be the way to go. And then there's the concern, and there was an NIH consensus conference about this. Why not just universally supplement? Why not just have every country say, look, let's give everybody, all pregnant women, iron? And the answer is because there are a lot of women that aren't iron deficient, and there is a risk to giving iron. So folks are hesitant to supplement iron sufficient people, but I don't think there's any question in the literature that you do treat people who are iron deficient.
Yeah. Okay, so Georgieff has a question. Georgieff, so you say mother with no iron deficiency and receives iron supplement, is it harmful to the baby? Great question.
Yeah, so not harmful to the baby, not by any study that I'm aware of. The concern about iron, and I can point you toward an issue, a special issue in the American Journal of Clinical Nutrition in 2017. That was a summary of this consensus conference at NIH about should we be supplementing iron sufficient pregnant women. And there's an article in there about an increased risk of diabetes in pregnancy. It's unclear what those mechanisms would be that would cause that, but there's still that issue out there that makes everybody, including our preventative task force in the US, unwilling to definitively say everybody should be supplemented with iron. But I'm aware that that can affect the mom and, I suppose secondarily the fetus, but not a direct effect on the fetus. It's not going to iron overload the fetus.
Thank you. Wow, these are cool questions. Sylvia's our developmental pediatrician. So Sylvia, if you are want to unmask yourself, you're talking about iron patches. Wow.
I have not had any experience with
Sylvia,
that.
tell us more.
Yeah, tell us more about that. Please feel free to unmute. And
are you able to unmute, Sylvia? I don't have a microphone. Good one.
I gotta look that up. To be honest, I have not heard of that. If anybody else in the audience has, let me know.
It sounds like a very, very good, you got patches for everything, right? Fentanyl. Yeah.
That's right. Yeah, that's an interesting
Well,
idea.
you can type on the chat pane. Iron patches are so online and not FDA approved.
Wow. Okay, I'm gonna have to check into that. So just to put a finer point on that little thing I said at the end, it's actually kind of a big thing. So there are now several studies that show, this was already known in adults, but several studies in young children, one by Michael Zimmerman, one by Nancy Krebs, that show a shift in the microbiome of kids that are babies that are supplemented with iron. And it really does look like the system was set up, if you will, that a baby accretes, or a fetus accretes a large amount of iron in utero, then gets delayed cord clamping, we call that appropriate cord clamping now, and grows at a very moderate rate on a low iron diet postnatally, right? There's enough iron in those stores to last for 4 months. By 4 months, a baby is much more immune competent and much less likely to be at risk of that changed microbiome. So just to give you an idea of some of the what we call siderophilic or iron-loving bacteria in the neonatal gut, they include E. coli, they include Salmonella. Lactobacillus, which is, of course, the beneficial one, has a very low iron need. So it looks like the system is built not to be supplementing kids enterally with iron, at least not too early.
So that's what Srini is asking. Do you use IV for the kids who can't eat?
Yes, we do. That's pretty standard therapy. And I think all of us who are perhaps a bit more on the veteran side, experienced neonatologists, were raised in an era of, IV iron is just terrible reactions to it and so on. But in fact, the new IV iron preps appear to be very, very safe with very minimal reactions. There are a couple trials that I know of in NICUs that are going on of IV iron as opposed to enteral iron in kids who don't have gut disorders, literally as the way to maintain iron status. And we'll see what they show. But yes, I think for a long time now, those who are strictly NPO for a long period of time or have gut issues, yes, they get IV iron.
So I've got a reference to transdermal iron that Sylvia was talking about.
I like
and
it.
some peds are using iron patches as a resource for kids who can't eat or
Nice.
don't want to
Nice.
eat. That's a
Nice.
great one. Sylvia, thank you.
Yeah.
Wow.
I'll have to take a look at that. That's great.
Yeah,
I
definitely.
wonder if it stains. I would think it would stain the arms. The parents would be very upset about that.
Well, I'm looking it up. It's not bad. It's like $28 from Amazon. Yeah.
Huh.
Gosh. Ooh. Thank you, Sylvia. We need to talk more.
Yeah, really. I love it. I love it. That's great.
Yeah, maybe I'll put you in contact with Michael. Set up an email chain and see what you guys can come up with. Thank you,
Great.
Sylvia. So the next question is, what's the consensus on OnePlus? Sorry, don't know your name. What's the consensus on withholding iron supplementation in NICU preemies who are positive? Oh, for who are infected. Yeah. Okay.
Yeah. There is no consensus there, but there are a lot of units that will stop the iron until the sepsis is under control. There's also units that stop iron therapy when they do blood transfusions too. None of it's really been studied rigorously. I think if you remember to restart the iron, it's not a big deal. Iron is the type of thing that is, like, because you have such large stores that you can afford to go, to be conservative and go a few days without iron or whatever. It's not going to hurt the kid. It's certainly not going to hurt their brain. So, if you feel that you want to stop the iron for, like I said, for transfusion, or you want to stop the iron for presumed sepsis, I don't think there's anything wrong with that. Just remember to restart it.
All right. From Gayatri in Perth, is it possible the association of iron deficiency with autism and schizophrenia was related to problems with abnormal gut microbiomes?
A great question.
So, we are good today. We're on fire. Well,
Lisa, it tells you everybody who stayed awake. That's my criteria for at least people we're obviously processing it too.
Is it okay for me to put your email on anyway? Just in
case.
Yeah,
sure.
Yeah, thank you. Okay.
So that's a really interesting question, because people are studying the gut-brain axis. In fact, we just hired a person in our division of neonatology to do that, and I thought she just studied, microbiome and brain. Turns out she wants to study iron deficiency in microbiome and brain. So maybe she comes up with that answer. The study that links with schizophrenia is the epidemiology study is from the Dutch hunger winter, where moms, it was a very short period, but it was a severe undernutrition, including iron deficiency, which they were able to pull out statistically. And obviously that's in the 1940s. We have no idea what their microbiome was. The autism link is actually by a woman by the name of Rebecca Schmidt, who is out at the Mind Institute at University of California. She might have an opportunity to look at that question. I think that's a good one. There are certainly, in autistic patients, which I see in my center, my institute, there are enormous amount of gastrointestinal issues as well as the neurodevelopmental issues. So it really begs that question.
Thank you. We are sort of like over time now, but I've put Michael's email on the chat group, or you can just contact us if you want to contact him for any more questions. Great talk. I'll put you and Sylvia together. You can talk about transdermal patches. I can think of many uses for that one. For a lot of children, it's wild. Yeah.
Adolescence, by the way, is a huge, the third big time period of iron deficiency, right? So we talked about the fetus, then there's the toddlers. We all know about those, but the third one is females, and especially female adolescents with their onset of menses, right? So they're the ones that will eventually be the women heading into pregnancy with marginal iron status, right? And so it's a great question and maybe a great solution.
Amazing. Thank you so much, Michael. Well, thank you for staying up and speaking to us on the other side of the world. Get your coffee. Get your coffee when I see you in PAS.
That sounds good. Anybody else who will be at PAS, please stop and say hello. I'd love to meet you.
Brilliant. Thank you so much, and have a good evening. Thanks, everyone.
All right. Take care, everybody. Bye-bye.
Goodbye. Bye.